Brilliant. So I've just seen that the record has started there. Are we waiting for more people to join?
So we've got Monica, Elspeth. Let's just have a quick little glance. So I think most people do today. Very you can't. I think Susan could though. I'll just check. Couple of minutes and then start.
Yeah.
And we can always record it. Thanks, Liz. I've got it's working at the moment.
Right.
Undoubtedly because all of the kids have gone away now, so the bandwidth is okay.
Yeah. Can I say apologies? I am going to have to duck and dive into this meeting because I'm already in a meeting that was supposed to be an all day. But John was asking if I'd be able to record, so I will keep you on in the background, but I won't record it. Okay, John? Yeah.
Cool. Thank you.
Okay.
And I've just had a message from our second scientist, Nikki, who's just having a little trouble logging in. So I'm just going to email you her email address, Mr. Hale,
okay.
And ask if you can forward on the invitation to her. And how many classes have we got in here with us today?
I think it's 2 or 3.
Great. Okay, that's brilliant.
There's Nikki there. Sorry. Cool.
Hi. Oh.
Hi, Nikki.
Hi. I can't see you, but that's.
Oh, there you are. Can you see me, Jack? Am I on?
The Matt and I can see
Okay.
you there. Excuse us,
Okay, great.
everyone. Our organisation is a Zooms organisation, so we're all a little. And I was just looking to make sure that I can share the slides as well. I'm going to control the slides from here. And yeah, then I'll just wait for you to tell me when to start.
Did you get a little message, Jack?
Yeah, absolutely. Yeah.
Yeah. Cool. So, Elspeth, if you can let us know when you're ready. Is Monica and April. Is anybody else expecting more students, or are we okay to start? I think, yeah, that's over to you then, Jack.
Fantastic. Okay, well, thank you all very, very much for coming along to our talk. I'm certainly very excited to be here talking with you, and I've got two of our scientists here as well. I'm going to start sharing our screen so you can see our lovely logo. And let me get that. There we go. So for everyone on the call, my name's Jack, and I am from a science research project called the Darwin Tree of Life. Now, my role on the Darwin Tree of Life is as public engagement coordinator. So I get involved in lovely interactions like this with all of you and with projects similar to the fantastic daffodil project that you've been part of. But I've got two other people with me who are involved in other parts of the project too, and they're going to tell you some more about what they do as part of the Darwin Tree of Life. What our aim on the Darwin Tree of Life is to sequence everything. So just like you've been creating genome sequences for your daffodils, we're doing the same for all of the different species in the UK and Ireland. So this talk here we've called Daffodils, DNA and Darwin. We'll be talking a little bit about those, but more we'll be talking about what it is we do on the Darwin Tree of Life and what the roles of both scientists and non-science staff on the Darwin Tree of Life involve. So at this point, I'm going to hand you over to my two colleagues, so Dr. Nikki Chapman and Dr. Max Brown. Those titles sound very serious, but I promise they're not very serious at all. And Nikki works in our legal team. So Wildlife and Environmental Law Advisor is her title. She'll tell you exactly what that entails and some of the ethics and legal questions that come her way. And we've got Dr. Max Brown, again, sounds very serious. He's a senior bioinformatician, again, a very serious sounding title. He's going to unpick for you what that exactly means and tell you about some of the science that he's been doing. He's been doing a research project which is actually quite similar to the work that you've been doing with daffodils. And we're going to be talking for about half an hour. And then after that, there's going to be plenty of space for your questions. And I know that we've already received some questions from the class, and we were astounded at just how challenging already those questions were. Clearly, you all know lots and lots about this subject. So I'm going to hand over to Nikki, first of all, to start us off. So over to you, Nikki.
Hi, yes, thanks very much, everyone, and good to see you. So, yeah, and thanks, Jack, for introducing me. In short, I basically advise on any protected species or protected sites. So that's what I do.
Max, what about you? Unpick that senior bioinformatician for us. What does that mean?
And yeah, hello everyone. I'm a senior bioinformatician, which basically means I look at DNA sequence data that's produced by the Darwin Tree of Life project, and I write software to try and understand what's going on, and I use other people's software to unpick and really dive deep into the genomic data that we produce.
Thank you very much, Max. So we have used that word Darwin Tree of Life already a couple of times so far today, but we've not really told you what it means. So here's what we're going to be going through on our talk today. We're going to tell you who we are, and you might think, well, you've already done that. We've heard it's Jack, it's Max, and it's Nikki on the call. Well, we're going to be saying who all the rest of the team are, because there's a huge number of people who work on this project, and who the Darwin Tree of Life project is. What does that mean? What do those words mean, and why have we chosen them for our research? We'll also be telling you what we do, and some of those things you may recognize from the experiments you've already been running in class. What is it that the Darwin Tree of Life is aiming to do, and how are we doing that? And then lastly, we're going to finish off with why. Why on earth should you try and sequence everything? What are these genomes for? And what can biodiversity genomics, which is exactly what you've been doing with your daffodils, why on earth should we care about that as a subject? And why might you want to do that as a job in the future? So I'm going to hand back over to Nikki now to take us through that who.
Thank you very much. Okay, who we are. Who is Wellcome Trust Sanger? Well, the Sanger is an institute that focuses on genomic and genetic research. We're a world-leading organisation for human and pathogen research, and we work with a wide range of organisations, so we're highly collaborative. We also train scientists in genetics and genome research. And more recently, we're spreading out into wildlife genome research. So we're defined by projects, and the Human Genome Project was a huge global project, and it involved many global partners, and Sanger was the UK partner. So what did it do? It basically sequenced the genome, which is the entire length of DNA, and it came up with answers for that focused on nutrition, medicine, and applying vaccines to diseases such as SARS. So it's a big project. Okay, so what projects do we lead on? Well, one of them is the Darwin Tree of Life, and the context of this was from Charles Darwin. Charles Darwin is a famous evolutionary biologist, and he produced a theory of how life or species are connected across an evolutionary tree. We're taking this and putting this into context for wildlife across Britain and Ireland. And so together we have Darwin Tree of Life project. The Darwin Tree of Life is a really ambitious project. We're hoping to sequence over 70,000 species. So putting this into context, how many species do you see in your garden any one time? 10, 20? We're doing 70,000. That's a huge number. As we said before, we're highly collaborative, and we work with quite a few organisations, and some of them are on this slide here. Some of them you might be able to recognise. For example, the Natural History Museum, Kew, at University of Cambridge. You may have heard of these before. You might have even visited them. Okay, we know where the University of Cambridge is. Where's the team based? Where's the Tree of Life project based? That's based at the Wellcome Geno campus in a place called Hinxton, which is in Cambridge. I can see on this Google map exactly where we are. Okay, this slide provides an overview of our campus. And the campus basically consists of loads of buildings. And we have loads of buildings, we have lots of research, and we have about 6,500 staff. On site, we also have a conference centre that's seen in the blue round disc roof. We also have a wetlands and a woodlands on site, and a place called Hinxton Hall. So it's a large place. And we even have a couple of restaurants. Okay, bye. Okay, our teams are also diverse as well. So this slide depicts an insect-based team, and there's lots of different people within one team doing different roles at any one time. It could be outside in the field, or it could be inside doing various different parts of the whole work that goes from the start to the end of the project. So we have 200 staff, the Darwin Tree of Life, but wider Sanger has about 1,000 staff. So this could be you. If you have a career in science, you can have different roles across many countries. Okay, so how have I got here? So I started off about 12 with a love of animals. I had loads of pets. So I was always going to be animal focused. I also lived by the sea, and I really loved rock pooling. I initially wanted to be a vet, but I did some work experience and it just wasn't for me. But what I did see about at the age of 16, I saw a program by David Attenborough on coral reefs. So I decided to become a marine biologist. And to get there, I studied biology, chemistry, and economics. And I went on to do a degree in marine biology. At the same time, I started scuba diving. Some of it was quite cold, it was in the UK. And later on, I did a lot of voluntary work to get some experience to get a job. So I did a lot of voluntary work surveying coral reefs. I then worked for the government, managing marine projects. And later on, I managed terrestrial projects. A bit later on than that, I managed projects overseas. This final picture is me in a chimpanzee rescue centre. So I was working to protect chimpanzees, elephants, and a weird species known as a pangolin against illegal wildlife trade. And then I returned to the UK and I put all these different strands together and then worked for Sanger. So I've taken a few twists and turns, but the themes stay the same. So I think a bit more about Max would be interesting.
Let me unmute. Hi everyone, thanks Nikki for the introduction. I started out with a love for insects. So throughout my childhood, I was always interested in entomology and especially beetles. And I enjoyed biology at school and chemistry, and didn't really get on with much else. But when I got to university, I studied dung beetles for my thesis. But I realized I knew nothing about plants, and lots of people don't, I guess. So I sort of switched for my PhD and looked at these little parasitic plants you can see in that middle photo. And more recently, I've moved to the Wellcome Sanger Institute, and I'm working on plants still, but things more like this ancient tree on the right.
Just before we move on to the next section now, Max is going to take us through some of the scientific processes and how we collect our genomes. But I'm just going to review what we've heard there. So you've heard a couple of different terms. You've got the Darwin Tree of Life. That's our research project aiming to sequence, that mysterious word, all of these genomes of all of these species, 70,000 plus species across the UK and Ireland. You've also heard Sanger, or the Wellcome Sanger Institute, and that's where the three of us are based. That's where we do our work, and that's in Cambridgeshire. The Wellcome Sanger does all sorts of other different things as well, and Darwin Tree of Life. But how are we going to go about collecting all of those genomes? How are we going to find those species in order to collect them? I'm going to hand back over to Max to talk us through that.
Yeah, so as Jack said, how are we going to do it? What are we going to do? And I'm going to go through that now. So there's this term called biodiversity genomics. And what does that mean? It means it's a way of understanding the diversity of life by looking at DNA sequence data. And we can begin to do this with the Darwin Tree of Life project. But first of all, I'd like to tell you about what a genome is. I'm sure some of you know, but maybe not all of you. So the genome is composed of 4 letters, the nucleotides A, T, G, and C, and they're string out in a really, really long string, basically. And these these letters can be grouped together in genes, which are basically just sections of DNA. And you can think of these sections of DNA being grouped on very large molecules called chromosomes. And you may know in humans that we have 23 pairs of chromosomes. And then the entirety of all of these chromosomes put together is the complete DNA code of an organism with one copy present in every nucleus, and that's called the genome. And the first animal to be sequenced was this little worm called Caenorhabditis elegans, which is a model organism for many different kinds of biology. But it was especially useful for understanding the genomes of animals. And you can see here that the genome size is 100 million bases, which you think is pretty big. It's about 9,600 pages, and if you print it out, you know, it's in those series of books there. But there's a diversity of genome sizes. For example, humans have 3,000 megabases, so that's 3,000 million bases, 3 billion letters of code. And you can see that huge stack of books on the left there that that lady's looking at is printed out the entirety of the human genome. But there's even bigger things like mistletoe, which you would think, oh, that's not a very big organism, but it's got a huge genome, 100,000 million base pairs. That's a trillion letters. It's almost unimaginably large. And we are going to try and sequence all of the diversity. So what do? We first have to go out and collect these organisms. So there are teams of people within the Darwin Tree of Life who go out and they sample organisms from the environment. This is everything from beetles to fungi to plants to single-celled marine animals and algae. And then you have to process them. So you freeze them in liquid nitrogen to preserve the DNA, and you ship them to places which can do the sequencing. And then you extract the DNA and you load them onto sequencing machines. And then number 3 in this pathway here, you can see that people are beginning to analyze these genomes now. So they're just coming through in the pipelines at the moment. So how do we collect? We go out into the field. We have to plan what we're going to get. We have to get legal permission, which is what Nikki works on. We've got to actually find the species out in the field. And this is usually done by experts in different groups of organisms. So we may have insect experts which go out and look for specific kinds of insects. And then we also use the DNA of the organism to check that it is what we think it is. So we check a small section of DNA called a barcode against a database of what we know of known barcodes which relate to species, and we check that we have the organism that we think we have. So we're just going to play a short video of collections at the Benay National Nature Reserve, which is in Scotland. Does it have sound? So you can see some of the techniques that are used in the field, such as sweeping, which is a really useful way of finding small organisms. And then you can check what's in the net and use these devices called pooters to suck them into small tubes. But you can also just root around in the ground, and that's a really good way of finding organisms as well, such as caterpillars and lichens and mosses. Suction sampling is quite a specialized technique which uses a big suction device to sample organisms which occur sort of just beneath where you'd be able to collect. And you can collect many, many different kinds of, especially insects in this way. Thanks for that, Jack. So next we have the process of extracting our DNA. So you can see there's a scale here. We have large, medium, small, and very small organisms. So we have to be able to extract DNA from every single one of these. But the main protocol goes that you crush the organism, you dissolve the DNA into a buffer or a solution, and then that is what is used to do final extractions and what the lab teams use to put onto the sequencing machines. And here's another short video of some of the techniques our scientists use in the lab. So sample sorting is a really important skill to be able to distinguish different types of organisms in a large sample. You can see that some of our scientists are using liquid nitrogen there, which is, I think it's nearly minus 200 degrees, so it's really cold. And that preserves the DNA. And then you can use things like power meshes to crush the organisms and extract that DNA. And you can see that there are different colors in the solutions from the different organisms. But in the end, at the end of all of this, there should be a solution which is clear, which contains the DNA. Centrifuging draws down all the heavier material to remove all the heavy stuff which isn't DNA. Magnetic beads stick to the DNA so we can remove it from the solution. And it's really clever. These magnetic beads stick to the DNA, and then you can use a magnet to draw all of the DNA to the side. And safety first. So then, once we have that DNA, we can load it onto a sequencing machine, and there are loads and loads of sequencing machines at the Wellcome Sanger Institute, and they sequence a variety of lengths of DNA. We usually use machines which are able to sequence very long stretches of DNA, because that's useful for later on when we're trying to reassemble the genome. And here we go. This is some of our scientists who take those text files which come out of the sequencing machines, which are just those 4 letters in huge files, gigabytes of data, and they're stored on these servers. And then scientists go in there and they use software and programs and algorithms to piece together these long sections of DNA. And it requires a huge amount of computational power because these files are so big. And you can imagine the larger the genome is, the more memory and computational power is needed to assemble the DNA. And what we're really looking for is, if you look on the right-hand side there, you can see that the DNA is assembled into chromosomes, which is what the Darwin Tree of Life aims to do for every single species. And then a really important part of what we do is sharing our data. So we do that immediately upon our curated final assembly. We release it to the world so researchers all over the world can use and analyze our data. And as of the 20th of October this year, we have nearly 200 species which have been submitted. Most of these are insects. And over 1,300 species have some kind of data. So we're really ramping up the project and its ambitions, and we're getting to sample the tree of life in its whole. So just an overview, we collect the organisms, we process them by extracting the DNA. We then read those DNA fragments using sequencing machines. Then the computers and the servers assemble these genomes, and then we share it to the world. As I said, I'm a bioinformatician, so I work with the sequence data, so I'm right at the end of the pipeline. But I'll hand over to Nikki now to tell you what she does and delve further into some of the aspects of the project.
Okay, thanks, Matt. Yeah, I provide advice right at the beginning. So prior to the team going out to collect samples, I advise on what to collect, if it's protected or not, and what site they're sampling from, and any rules they have to abide by legally. I also provide a second bit of information just prior to samples coming to Sanger, because what they intend to collect and what they do collect can differ due to what's available. Okay, so why do we do this and why should you care? And they're really good questions. So you've heard a lot about the processes and what we do. So why do we do this? So the research supports conservation and information on biodiversity. So we're going to have a quick quiz because we've done a lot of speaking and we want this to be interactive. So the next couple of slides, we're going to guess the protected species or the protected site from the pictures. Okay, we have 3 pictures here. One you may recognise. And what I'd like to know is which of these plants is protected by UK law. Is it A, the daffodil, B, an orchid, or C, daisy? And how we're going to do this, we're going to ask for hands up. I'll give you a couple of minutes. Okay, hands up if anybody thinks it's a daffodil, is legally protected by law. Hands up if anyone thinks it's the orchid that's protected by law. And hands up if we think it's a daisy that's protected by law. Okay.
Looks like we've had quite a few votes in for orchid so far. Certainly I can see 2 votes for orchid. I wonder if there's any other votes in there, but yeah, Nikki, I'll hand over to you. What's the answer?
Well done. It is the orchid, so it's illegal to pick it. It is available in the wild. We have some rare plants in the UK. Okay, we try one more. So protected sites, and these can range. So in this slide, we have 3 genuine protected sites. 1 or more of them can be protected. So I'll just go through them. A badger set, that's a home of a badger. A bat roost, that's a home of bats, or woodlands around the UK. So this one, I'm going to ask which one is protected, or do you think all 3 are protected? So it's a little bit more complicated. So I'm going to say hands up if you think it's a badger set only protected. Hands up if you think it's only the bat roost protected. Or hands up if you think woodlands are protected. And the bonus one, hands up for those who think all 3 are protected.
Looks like we've got votes in for all 3, Nikki. What's the answer?
Excellent, well done. That's really good stuff. All 3 are protected. The first 2, badgers and bats, are protected species, and we have a range of woodlands that are protected around the UK as well. Fantastic stuff. Okay, so we're looking at the conservation side. We've also got very interesting wildlife, and in the next couple of slides, we're going to continue with the quiz. Okay, so here, we want you to look visually at the species and see if you think they're related or not. Okay, so we see the shark. What do we think is the most closely related to the shark? Do you think it's A, the dolphin, or B, the tuna fish? So hands up for dolphin. and hands up for tuna fish as being the most closely related to the shark. Oh, got some more.
Yeah, we got one vote in there for tuna fish. Didn't see anything from the other class. Have we got a vote in from the other class? B, also tuna fish from the other class.
Oh, this is a good audience here. So yes, you're right. It's tuna fish. The tuna fish and the shark are both fish. The dolphin isn't a fish, it's a mammal. It breathes air. Fantastic. Well, let's try a little more tricky one. Okay, next slide. So a hyrax is a small mammal. So do we think this is most related, closely related to a rabbit, A, or B, the elephant? Hands up for A, rabbit, and hands up for B, elephant.
Okay, we've got a split here. So we had one vote from one class for A, rabbit, and we had the other class had said B, the elephant. Who is correct, Nikki?
Well, this one is a tricky one. So it's the elephant is most closely related, and it's to do with the nasal structure. And it really demonstrates you can't tell the difference or the between the relationships between species just by their external features, sorry. Okay, so because it's tricky, we use genetics to identify the relationship and the closeness of the relationship between different two species. And so how do we do this? We look at the DNA. And within the DNA, you see certain bands. And in this slide, we can see those bands in grey and blue. So we have human banding pattern. We have a chimpanzee banding pattern. We have a gorilla banding pattern and the orangutan. So I'm wondering if you can have a little look and see which one of these, have a little think, which ones you think are most closely related by looking at the banding patterns. Okay. So if we move on to the next slide, we'll find out. Okay, so on the left-hand side, we can see the human and the chimpanzee are most closely related. That means their banding patterns are more similar than the gorilla or the orangutan. Quite unexpectedly, the orangutan is the least similar, but we can look at the banding patterns and see the distinction. Again, these branches on the left-hand side, they are called lines or branches. They make up of what we call the tree of life. So that's the sort of image we see. Okay. Okay, this one's a little bit more tricky. It's the last one. So, which is mostly related to a mushroom? Is it A, humans, us, or B, a dandelion? And this is the last hands up. So A, hands up for, are we most closely related to a mushroom, or dandelions more closely related to a mushroom?
Both votes for dandelions there. Oh, we've had one second vote in from the other class, which says A, humans. So again, we're split. One vote for humans, one vote for dandelions.
Okay, again, this is a little tricky outsider one. So we'll see by the next slide. Okay, worryingly, we are most connected to the mushroom. And so that's a take home. So the mushroom actually isn't related to plants, although again, it could look like a plant. We are more related to, and those lines at the bottom show the relationship between animals and fungi. The lines are closer together between animals and fungi than fungi and plants. Again, the lines reflecting a tree of life. Okay, and this picture shows a colossal tree of life. It shows the tree of life and the branches from the beginning of time to now. So from when the first bacteria started, if we look around the outside, you get bacteria all the way around to mammals. And that's how life evolved. And the little branches will have several hundreds of branches, but that's all the species in a relationship too. And that shows you the tree of life. I'll hand you back to Max.
Brilliant. Thanks, Nikki, for that. I still find it incredible that you can compare sequences between organisms, and by counting the differences in bits of DNA, it gives you an incredible overview of the tree of life. We can really dig down and look at really ancient relationships between organisms in this way. So this is super powerful, but it doesn't answer why. Why are we interested in this, and why you should care? So looking at the genomes are going to transform the way we do biology. We'll be able to understand what different genes and regions do. So we can scan a genome and we can zoom in to a specific region and we can say, what does this bit do? We can do that in lots of different ways. We could extract that sequence and compare it to other sequences and see what its function is. Or we can do mutation studies where we try and knock out that gene and see how it affects what the organism looks like or does. And it allows research into new areas. At the moment, we still have a really strong bias for genomes that are present in public spaces for vertebrates and mammals. There are far fewer invertebrate genomes like insects or even sponges. So one of the things that I've been looking at in one of my research projects in collaboration with different organizations around the UK is investigating apples. And you may not know it, but there are thousands and thousands of varieties of apple in the UK alone, maybe upwards of 7,000. And we've sequenced a bunch of different cultivars from Scotland, England, and Wales, and we are trying to understand how they're related. So how Nikki was saying with those trees, you can build those trees by looking at the DNA of apples as well. Where do these sequences differ? We don't know. Why do they differ? Again, it's an important clue, and answering these questions will help us understand the origins of apples more clearly, and understand where parts of the genome influence parts of the apple, which you can imagine is super important for commercial apple growing, who desire things like apples that don't bruise easily, or longer fruiting time, or any of these sorts of things. We also want to save ourselves. We all know that the climate is changing, and food production could be modified by looking at genetics and selecting for genes which are better suited for high yielding crops. We'll be able to discover new drugs. Many drugs come from plants and fungi, and there are thousands and thousands of drugs out there waiting to be discovered, and we could discover those by looking at the codes in DNA and trying to understand whether sequences found in the production of interesting compounds in other plants or fungi or animals are found in the one you're looking at. There are many, especially organisms like algae, which can produce things like oil, and again, trying to perhaps genetically modify or look at the genomes of these organisms will help us understand how we could produce that. And of course, nature has been doing this for billions of years. We've only been around for just a few million. So nature is the place to look. So we also want to save more than just us. We don't want to have this total centric view, and that is why we do the project like Darwin Tree of Life. We want to understand how organisms respond to climate change so we can save these organisms, or perhaps try and save environments in which these organisms live. And we can understand and track biodiversity of invasive species, so we could see how and why certain invasive species are able to flourish in certain environments by correlating the environment in which they live with their genetic code. So, can you sequence everything? We're trying to. And so we'd just like to say, in conclusion, right now we have a huge library, and we know that there are these different life forms out there. And so the next step is to try and fill this library with the genomes of all the species on the planet. And we're going to start with the UK, but eventually it will be the planet. And then the job isn't done, because we have to read every one of those books, and we have to understand every book in the library. And we want to find all of the amazing transformational knowledge that they contain. And that isn't just a job for me or Nikki or Jack and us at the Darwin Tree of Life, but it will be a project for you as well as you progress into the world. So thank you for listening to us. And do you have any questions? We'd be happy to answer.
So thank you very much, Max and Nikki, for talking us through all the things there. We're very happy to stay on to answer any of the questions in either of the classes that are online with us today. That could be some stuff from that first section, the who, you know, some more about either Nikki, Max, or myself and our journey into science, the people that we work with, the place that we work, that Sanger Institute. Yeah, thank you very much, Elizabeth. Thank you for that. It could be about that what. I know that lots of you on the call will have experienced some of that what already in the daffodil experiments that you've been doing. You might want to know a bit more about our processes here. Maybe that's to do with the collection that Max mentioned, getting out there into the field, what we're collecting, how we're collecting it. Might be about those lab processes and how it is that we process those samples, or it might be what happens afterwards. It might be those sequences that are generated, some of the computer power that's put into that. And Max can answer those questions. There's also loads in there for Nikki about the ethics, I suppose, or you know, the how we manage some of those rarer samples, you know, when we have a rare or an endangered species. Here we've got a great one from Mr. Hale's class. How much will the Darwin Tree of Life cost? That's a big question, isn't it? So I can give a little bit of insight before I hand over to either of the two of you. Max there's got quite a surprised expression on his frozen screen, so I hope he's got a good answer too. I don't know if you remember that early slide about the Human Genome Project right back at the start. So that was this big project that took place about 20 years ago. The cost of Darwin Tree of Life to sequence everything in the UK and Ireland, and then this bigger project, which we haven't mentioned here, but it's called the Earth Biogenome Project, that's sequencing all of the species in the world. They realized that doing that now is the same cost as doing that Human Genome Project was 20 years ago. And that's all about progress, isn't it? That's like when you see those pictures of mobile phones 20 years ago versus mobile phones now, it's as the technology progresses, the price comes down, and we're able to do this big research project. Max or Nikki, I don't know whether I've given a bit of a vague answer there because I don't know what the actual total budget for Darwin Tree of Life is, but can either of you be more specific than I've been?
Oh, it's quite difficult. So you've got staff costs, you've got equipment costs, and it's an expanding and growing project over 7 years. So I'm guessing it being millions. altogether? I don't know. Max?
Yeah, that's really tough. It's a lot. I mean, just the sequencing alone is a lot of money. You know, perhaps a few £1,000s for every genome that we're doing. So you could multiply that by 70,000 and you've got a figure just for the sequencing. But as Nikki said, you've got staff costs, equipment costs, sampling costs. Yeah, it's a lot. Try not to think about that.
So it's a big number. All of that comes at the moment from a grant from a place called the Wellcome Trust. You might have seen beady-eyed students watching will have spotted Wellcome written on here. You might think, hang on, that's spelled in a bit of a weird way, WELLCOME. There's one too many L's in there. Well, that's because it's actually a person's name. The Wellcome Trust was made from the bequest of a man called Henry Wellcome a while ago, and so that's where the money's coming from for it. We've had a good follow-on question to that, though. So if it's going to cost this huge amount, why is it important to sequence a genome of living organisms? And that one was from Hannah. So I think, who feels like they're in a good position to answer that one?
Well, I can give part. It's almost a two-part question. So I can do it from the wildlife side, but there's other reasons too. So from the wildlife side, it's really important because we want to undiscover some mysteries. Are certain species related to others? Are they very different? And this will inform conservation in the future. So at the moment, when we have a protected species, we have things called conservation management plans, a series of steps to take to protect species. And we do that on the basis making assumptions about species that they're related and their needs are the same. And so we need to get information on that. And also, some species may or may not be resistant to climate change. So it also unpacks some understanding about that. What is the impact of some things that can come in the future on species? Can we predict that and mitigate that? And I don't know if Max has anything to add to that.
Yeah, I mean, that's a really broad question. I think, Nikki, you talked about some of the practical implications for organisms, but I guess there's all of the sort of blue sky thinking or testing evolutionary theory, which you can do with many different genomes. And another huge important part is that we can't research everything on the Darwin Tree of Life, but if we make these genomes available, people can use these genomes and as a reference, and then they can sequence their many different individuals of the organism of their interest, and then they can start to do population level studies, so compare many hundreds of individuals using the genome that we've produced, which I think is going to be of huge importance to the world for biology. So yeah, I hope we've answered some of that question.
Yeah, thank you very much, Max. So just to add to that, we mentioned about sharing those genomes. Those genomes are shared openly for everyone. So anyone listening to the call today could log on to the Wellcome Open Research portal, which is online. If you search Wellcome Open Research, and there you can access all the data for this project. So you'll have seen some of the DNA sequences you've got out of your daffodils. You can go in there and see the ones that we've already released for detail. We've got a couple more questions, so I'm going to keep us speeding along. Have any new species been found in the UK from sampling for Darwin Tree of Life? I'm going to give you both a little bit of time because I had a lovely story on this from Mark Blackster, who's our lead investigator on the project. This wasn't from Darwin Tree of Life sampling because we're quite new as a project. So we've, I don't think, found any new species, although the two on the call can correct me. But this same sort of genomic sequencing on a project that Mark was working on earlier discovered a new species of worm just in the compost heap on the research site. And they saw these genomic sequences, they tested these different worms, they saw that they looked like different species. They got a load of worm experts in to look at the worms. And when you separated them using their genomics, the experts could say, yep, that's one species, that's a different species. But if you mix them up together and then ask them to pick out which was one species and which was the other, they weren't able to do it. So there you can see how the genomic data can help us understand different species in a way that actually it's very, very difficult to do, or sometimes impossible to do just by looking at these species alone. And can either of you give me an advance on new species found on the Darwin Tree of Life data or on similar biodiversity genomic projects?
I can say something. So when we sequence the DNA of an organism, say we go out and sequence an insect, we're not just sequencing that insect, we're sequencing everything that is associated with that insect as well. And for example, in insects, there are parasitic bacteria and all sorts of microorganisms which can only grow and live with that particular species of insect. And we have found lots of these, they're called endosymbiotic organisms associated with insects. And many of those will be new to science. So it's kind of, it's a bit different to finding a new species of, mammal or something in the wilds of the UK. But there's plenty of stuff to discover, just even within one insect. So that's what I'll say.
Well, I'll move us on to our next question here because we've got another one. So what is the most surprising evolutionary relationship that you have discovered? And again, we've got using Darwin Tree of Life on here, but I'd say just in general in your careers, say something about a surprising evolutionary relationship.
For me, I think it is the hyrax and the relationship with the elephant, just because of why it's related. Because they are so different and look so different, and the sizes are so different. So the similarity in those two, it was quite a surprise, and it's just not obvious when you look at them. But it's, yeah.
What can I say? I think because the question's a little bit vague, you can say, okay, I've got the organism. How is an organism related to another organism? But you can also go a step further and say, how are the genes in this organism related to genes in other organisms? And because you can compare genes, each gene has an evolutionary history of its own, which is obviously dependent on the species, but it doesn't have to follow the species tree. I know that's a bit mind-boggling, but for example, you can have genes which hop from one organism to another. So you have genes which hop from bacteria quite frequently into species of plants. And you can have parasitic plants which parasitize the roots of other plants, and you can find that there's genes which are being passed from one plant to the other. And so if you were constructing a tree of that gene that you found that was transferred, it would be more closely related to the parasitic plant than any other closely related plant at the species level. So those are really surprising evolutionary relationships, but quite difficult to sort of get your head around sometimes.
I can see we've got two more questions that are coming in, just because I know that we're getting towards the end of our time together. I will say that any questions we don't get to, we'll absolutely make sure that your teachers write them down and send them over, and we'll answer as many of those as we can, because I think there are some really fantastic questions already coming in. So we've got here, do you agree with the Darwin Tree of Life priority species? I love this question already. If not, what would you change? So this is great. So that's calling back to that slide where we say, we know we've got 70,000 species, right? Which ones are we going to do first? That's a really difficult decision to make, isn't it? one thing I would say is that actually on our website, there is a place where people can suggest which species they would like us to prioritize. Normally that might be researchers who want to find out a particular thing. So we sometimes listen to the community there. What would I choose to prioritize? The first thing I thought was I'd love to get a load more bat sequences on there because I really like bats. I think they're very cool. But it's challenging, isn't it? Because what we might end up doing there is just replicating these biases that we already have in our genome databases, because we go, oh, that animal's really interesting, so let's do loads of studies on that. There are lots of very understudied types of life form out there, and so we want to make sure that we're finding out all about the weird fungi that live at the bottom of the tree roots in your garden or whatever. So yes, really challenging. I'm sure, Nikki, what would you say to that? What would you change about our priority species?
Yeah, so there's two things here. Everybody's got their different area of specialism, which is great. So mine would be marine biology, so I'd be looking at all marine invertebrates as priority, just for personal preference. But it isn't always what we prefer. So it's like what is available at the time as well, and trying to get. So if we're in the field collecting, we might want a butterfly X, but on that day there's no butterfly X. So we have to be a bit fluid, and I think the priority is to try and get representatives of each group of animal. So the broad thinking is good. As it's field work and things happen, it might not just go to plan sometimes.
Yeah, and just because you mentioned that word there, can you give us an example of a marine invertebrate? What would be an interesting marine invertebrate?
I love nudibranchs. Nudibranchs are sea slugs, so they're very beautiful and pretty, and you get them in the UK, crawling across rocks on the bottom and having a big chomp on a bit of seaweed.
Great, thank you. Okay, two more brilliant questions here. I hope we can squeeze them in. Okay, so we've got, where are the cells taken from in different organisms that you would sequence? For example, hair, feathers, saliva. And that's from Abigail. Great question, Abigail. Max, let's go to you on that one.
So it depends on which group of organisms you're looking at. So from the question, I'm guessing you're thinking of mammals, and usually I think they take either blood samples or ear clippings, maybe, or something like that. So like sort of non-destructive, the things that aren't going to harm the animals in that way. But for other groups of organisms like plants, you can take the leaves of plants or the buds of plants, really good. And sometimes things are just single cells, right? So you have to just sample that cell. And some things are so small that you wouldn't be able to discriminate between the different kinds of cells. So, for example, a very small fly, which is only a millimeter or two long, you may have to sample the entire thing. So it really varies between organisms.
Yeah, and I can confirm we have had bat wing clippings, we've had fish scales, we have blood samples from mammals. We also have like tissue samples, muscle tissue samples. So yeah, it is quite broad.
Great. We've got another good question here. When is the genome good enough to be published? And that's a very important question that I know that we're still grappling with at the moment. Who wants to answer on this one?
Could give it a go.
Yeah.
When is the genome good enough to be published? So I guess the answer is when it is as complete as it can be given our standards which we set out. So we want each of the species that we sample to assemble into chromosomes. So we want to go from the start all the way to the end of every single chromosome. So historically there have been very fragmented assemblies where you may, for an organism that has 23 pairs of chromosomes like us, you may have been able to assemble the genome into 100,000 fragments. And so that's really not good enough for what what we're doing. So it's good enough when we have a high, what's called contiguity of the assembly, and when we think there's a high completeness of the genome. So does it have the genes we expect to see in a genome? And so you can look at these things called housekeeping genes, which are essentially the genes that almost every organism has, no matter what you look at. And we can say, does this organism we sampled have these genes? And if it does, it's probably pretty complete. So yeah.
Brilliant. Thank you very much for that, Max. I'm just looking at my clock and I can see that we are now beyond 4:30. Teachers who are in here, you tell me, do we need to be stopping and closing up now? I can see we've got another question, but I'm aware. Yeah, we need to wrap up. Okay, thank you very much for that. Will we please, teachers, do make sure that you collect in any questions that we've not answered, send them over to us, and we will get some answers for you for those. But can I just say a huge thank you, classes, for joining us today. You've had some really fantastic questions for our scientists, and we've really enjoyed speaking to you. Good luck with the rest of your daffodil project. It sounds fantastic, and I can't wait to hear your results from it. And we hope to speak to you again soon. All right, take care, everyone.
Bye. Thank you.
Bye. Thank you.
Great, I think that's everyone out. So we can hang up. Brilliant. Well done, both of you. I won't ask for the debrief on this now, but I'd love to speak with both of you soon on how you felt that went. Yeah, any kind of feedback on it. I thought the presentation went really well, and I thought those questions at the end were excellent as well. There were some really good things.
Oh, they were great
questions. Questions were so good.
Yeah.
I could have probably spent the whole talk just doing the questions.
That was great. Yeah.
Yeah. There were some nice bits of feedback from the teachers there just at the end, and I'll make sure anything that gets passed on from John or the others gets fed back to you both as well. But big thank you. And
thank you, Jack.
Yeah.
Thank you. Thanks for organising it. I love the cat coming in.
Yeah.
I love it when cats come in. I swear, oh my God. One member of our team has a cat. Cat always comes across in the meeting with a tail wrapping around the neck. You can see her trying to do this, and it's not having it.
Hello. All right. Well, take care, both of you. Have a wonderful Christmas if I don't speak to you before you finish. And yeah, looking forward to seeing you in the new year.
You too.
Merry Christmas.
Yeah. Oh my God, Christmas already. Take care.
Yeah, gosh.
Bye.
Bye. Bye.